In this study topic, we're going to take a look at basic Mendelian genetics. So think Punnett squares, those simple monohybrid Punnett squares, and those dihybrid Punnett squares, and then meiosis, how that all ties into the separation of chromosomes during that process. So as you remember, reproduction, whether it is asexual like in bacteria where they're just making copies of themselves, or sexual reproduction, this all involves the passing on of their genetic information with chromosomes. As you remember, Mendel did not know what chromosomes were at the time when he came up with all of his ideas. But as students in this generation, we can tie that all together and look at how meiosis and sexual reproduction actually helps to explain Mendel's monohybrid and dihybrid crosses. When we learned this the first time, we also looked at chi-square, which we reviewed a few videos ago, and looked at how when we do a genetic cross, if what we get is close enough to what we expected based on Punnett squares. So we'll be looking at those sorts of things in this video. What would questions look like on the AP exam? Here's a list of those types of things. So knowing monohybrid and dihybrid basic outcomes. So think a 1-to-1 ratio or a 3-to-1 ratio in a monohybrid cross. And then in the dihybrid cross, we looked at a typical ratio of a 9-to-3 to 3-to-1, right? Think of that corn lab that we did where I had you counting corn for days. So we'll look at some practice questions like that in this video. For number 2, what does crossing over do to those expected ratios? We'll look at that also. We'll look at meiosis again and review that. And then what's the difference between linked genes and non-linked genes? And if they are linked, what is autosomal linked and what is sex linked? And then what are our other types of inheritable situations? So think of that silly lab that you did with the aliens that you drew. Incomplete dominance, co-dominant, sex linked, all of those different types of inheritance patterns. And then specific ones in humans, the ABO blood group, and crossing parents to kind of figure those out. So we'll look at a lot of these in this video. Again, here is a beautiful list of terms that you should review and refresh your memory on, and then email me or let me know if there's something that you're not remembering from this list. So quick refresher. Let's review meiosis. So this figure here on the left walks you through the whole process of meiosis. Again, you won't necessarily need to know all of the individual steps. You'll just have to know the outcome. So what I did was I took this complicated diagram and, drew it out without the steps, right? So let's look at the first time. Let's pretend that our organism has 4 chromosomes or 2 homologous pairs. And if the chromosomes line up like this at metaphase 1, remember the homologous pairs separate. So the red one goes to this cell, the blue one goes to this cell, purple, and then green. And the homologous chromosomes have separated in meiosis 1. Meiosis 2, the independent chromatids, the individual chromatids separate from each other. So we end up with red in each cell and purple in each of these cells. And same thing's going to happen over here where the blues separate and the green separate. So this possibility is one way that we could separate or segregate the chromosomes into our gametes. Now, based on how these line up, thinking of Mendel's law of independent assortment, what if instead of the purple being on the left, the purple is now on the right? If we follow that through meiosis, whoops, I forgot a chromosome here, I forgot my blue one. If we follow that through meiosis, are we getting the same outcome in this way? And the answer is no, right? So that's the law of independent assortment. So based on how the chromosomes pair up and line up at metaphase 1 could give you different options in the gametes at the end. In this example, I didn't even show any crossing over, right? We probably would see some crossing over between the red and the blue chromosome and the green and the purple, and we would probably see even totally different combinations of genes in each of the cell. Same thing would be true for this example over here. So meiosis, the process of meiosis really comes in and affects our Punnett squares. That's really where Punnett squares get their basis. Here was a question from 2016 that if we were doing meiosis at the time, you probably could have gotten this, but since it's been a little bit of time since you've done this, it might take a minute to refresh your memory. I gave this on a mock exam a few years ago, and I was like, oh, they know this. And when I got the results back, they did not. So I really want to hit this question pretty hard and really focus on what meiosis is going on here and how it's actually playing a role with letters, right? So when we talk about genotypes, when we talk about Punnett squares. So take a minute, pause the video, read this prompt, and then what I'd like you to do is on the next slide, there's a little practice section where you can plan out your answer and try it. So pause the video, try this, and then when you're ready, we'll look at the answers together. Okay, so a couple things to pay attention to. They're telling you the P generation is this crossed with this, and they're producing an F1 generation. So if these are the parents, this parent can only give a big G, and this parent can only give a little g. So the F1 generation is going to be heterozygous for flower color. Also looking at plant height, parent 1 can only give this, parent 2 can only give this. So the F1 generation that would be produced from these parents would be completely heterozygous. This is a key thing to understand to help you with part A. Now we're looking at the F1 generation. So we're looking at this genotype, and we're going to use the diagram to follow the chromosomes through meiosis. And they give you some hints here showing you that the flower color gene is going to be located on chromosome number 1. So let's flip to the next slide that gives you a place to sketch out and think through your answer. And I'm just going to draw my circles in here so that we have these ready for when we need them. Okay. Now, the key thing that we looked at on the previous slide is that flower color is going to be located on the big chromosome, so on chromosome number 1. Let me flip to green here. So if our parent, our F1, was big G, little g, we know that this chromosome is going to be carrying 2 copies of the dominant allele, and this chromosome is going to be carrying 2 copies of the recessive allele. The small chromosomes are carrying the plant height genes. So this chromosome, let's just assume it's carrying the dominant alleles, and this chromosome is carrying the recessive alleles. Now you might be asking, wait, why are there 2 copies? If you remember, before meiosis begins, DNA replication happens and copies all of the DNA. That's where the 2 chromatids come from in each of the chromosomes. Remember, they're identical DNA to each other. So that's why I'm using 2 of the same, okay? So now, in meiosis 1, as we reviewed on this picture here, what was separating in meiosis 1? What could you call these things, the red and the green, or the red and the blue? We know that those are called homologous chromosomes. So down here, that's kind of what you want to be thinking about. These are going to be your homologous chromosomes that are separating. So if we're following this through, we know that the 2 big chromosomes are going to line up at metaphase 1 and then separate. So give me a second while I sketch this here. You're going to have a big green chromosome here, so big G, and you're going to have little g here. And the same thing's going to happen with your plant height. So we could see, let's do big D here, and little d here. What is produced? We now have haploid cells. and specifically we have 2 haploid cells. Okay, now let's follow that through meiosis II. As you know, when we look back at this figure, down here, what was separating the individual chromatids? So we call those the sister chromatids. So that's what you can fill in this box, the sister chromatids. And if we follow that through, this cell is going to get 1 copy of that. This cell is going to get the other copy. These 2 cells, we're going to see little g in one, little g in the other. And then we follow that through our blue. This is going to get big D, little d, little d. And what's produced here, we're going to end up with those 4 haploid. Now we can officially call these gametes. But thinking about the figure that we looked at over here, is that the only combination of chromosomes that we could get from this parent cell? Does it necessarily mean that we have to get the big G and the big D together in the same cell? And do we absolutely have to get the little letters together in the same cell? And your answer should be no, right? If one of these things lines up the other way, so let's instead pretend, actually, let me just do it in a different color. Let's do it in light blue. Let's say in situation number 2, this lines up like this. So the D's are switched. Okay, that's going to switch everything in meiosis I. And if we follow that through, that's going to switch everything in meiosis II. Okay, so what are we looking for in this? We're looking for the 4 possible combinations of letters that we could get. Okay, so this is the logic behind it. This is the meiosis that's going on in this question. But you might remember that when we were figuring out possible gametes, I taught you a quick FOIL method, right? So if we have big G, little g, big D, little d, we can FOIL this like you do in math to figure out our 4 possible gametes. So if you are able to just jump right to that FOIL method, do that. What I wanted to show you back here, and it looks really messy right now, is the logic behind that, right? So based on independent assortment, based on where those chromosomes line up, we can get 4 different gametes and 4 possibilities there, right? Just like we did over, oops, sorry, yeah. Just so we did over here, we got 4 different combinations of colors, okay? So for part A, they wanted you to draw the cells. So let me flip to this. This is what the answer would look like if you were drawing this out. And these are the letters that we just figured out with FOIL. So big with big, little with big, big with little, little with little. Okay, so that would have been 1 point getting that. Now, part B is asking you to now predict the possible phenotypes and their ratios if we cross an F1 individual with a completely homozygous recessive individual. This is just a Punnett square, right? So if we take the F1 genotype, which back here we figured it out to be big G, big D, little d, and we cross that, we would see a 1:1:1:1 ratio. So if you did your Punnett square, and this again is going to be a big Punnett square, that's where those numbers came from. Okay, so give me a second, I'll get that for you. Here we have our F1 parent that we FOILed out, and then they tell us we're doing a test cross with a completely homozygous recessive. So you could take the time and write all of those gametes out, but what do you notice? What are we going to get? All of these are going to be the same, all of these are going to be the same, and so on. And that's where they got these ratios for part B. Let's pause here because part C is going to take me more than 10 seconds, and I will pick up on a second video to finish this section.